The first speaker will be Dr. Fadi Bitar, and he will be talking about advances in molecular biology. Dr. Bitar.
Good morning. Thank you, Mr. Chairman. First, I would like to thank the organizing committee for inviting me, and I congratulate them on an excellent meeting. It's always a pleasure to be back in Riyadh among colleagues and friends. Over the next 30 minutes or so, I'll try to shed some light on the molecular basis of congenital heart disease and translate some of the basic science knowledge that we know into clinical practice in the area of congenital heart disease. As we all know, congenital heart disease is one of the most common cardiac malformations and the leading cause of death in the first year of life. Regardless of the major advances in the diagnosis and treatment of congenital heart disease, little is known about the etiology. Speculation about the etiology of congenital heart disease started in the 50s, 60s, early 70s, where the multifactorial inheritance theory was proposed, which includes an interplay between genetic and environmental factors as causes of congenital heart disease. However, the advances in the diagnostic molecular genetic tools opened the door to better understanding of the etiology of heart disease. Over the next few slides, I will talk a little bit about the major advances in the field of molecular genetics. And in the 50s, the DNA structure, the double helix DNA structure was discovered, and it includes 4 nucleotides: the adenosine, the cytosine, the thymine, and the guanine. And these are present in a specific pattern which bind the 2 parts of the helix, where adenosine will bind only to thymine and cytosine will bind only to guanine. In the 60s, the discovery of the triplet genetic code was available, also the sequencing of the DNA. And the triplet code indicates that a triplet sequence of the nucleotides in the DNA will translate to a triplet sequence in the RNA, and this will code for one of the 20 known amino acids which will constitute the protein, which is the major molecule that determines the function of a particular cell. In the 80s, the polymerase chain reaction was discovered. This opened the door for mega amplification of the DNA. One can produce million copies of a single DNA molecule, and this was described as the copying machine of the DNA. This particular tool opened the door for better understanding and studying of the molecular basis of various diseases, including congenital heart disease. However, the major breakthrough was in the year 2000, where the draft of the sequence of the human genome was available. Of course, the definite sequencing was available 2 years later. And again, the coding part of the DNA is about 1 to 2% of the total DNA. The coding part of the DNA will translate into the formation of RNA. These are called the axons. However, the non-coding part, which constitute about 98% of the DNA, which is called the intron, is a non-coding part. This will leave us with about 30 to 35,000 available genes that will code for specific diseases. So in brief, what's happening, the DNA will transcribe in the formation of RNA. The RNA, messenger RNA, will leave the nucleus into the cytoplasm, attach to the ribosome, and produce the protein, which is the molecule that is responsible for a functioning of a particular cell. So any abnormalities along these lines will lead to abnormalities in the protein production and thus abnormalities in a function of a particular cell. Now going back to the congenital heart diseases, one can relate congenital heart diseases into various abnormalities, including chromosomal abnormalities, which is associated with about 5% of patients with congenital heart disease, single gene defect 3%, environmental factors. However, 90% of congenital heart disease are sporadic and familiar cases with no well-identifiable cause at this point. What I will do in the next few slides, talk a little bit about chromosomal abnormalities that's associated with congenital heart disease, gene deletions, and mutations in some of the nucleotides that is reported in patients who have congenital heart disease. Remember that these three categories account only for 10% of all congenital heart disease. The later part of my talk will address the sporadic cases of congenital heart disease. All of us know that patients who have chromosomal abnormalities have higher incidence of congenital heart disease as compared to the general population. The very well-known example is trisomy 21. Probably 1 out of 2 babies with Down syndrome will have congenital heart disease. Turner syndrome, 35% will have congenital heart disease, in particular coarctation of the aorta. And trisomy 18 and trisomy 13, these patients have certain dysmorphic features and will have more than 90% association with congenital heart disease. So here we have a specific gene that in most of the cases will code for a specific type of congenital heart disease, and this genetic alteration seems to be specific rather than global. This group of patients, one can do the diagnosis by a simple karyotype. However, there's another group of patients who have gene deletion, and this is also associated with congenital heart disease. These cannot be identified in the regular karyotype that we order. So one has to use another technique called the FISH technique, the fluorescence in situ hybridization. This particular technique involves the use of a specific probe, a fluorescent probe that will attach to a specific gene on a particular chromosome. So if that gene is present, then fluorescent will appear on the slide. If that particular gene is deleted, then the probe cannot attach to it and the fluorescence will not appear. This particular technique is used to detect major deletions in major genes that include sometimes thousands and millions of nucleotides or base pairs. And one of these gene deletions that is well known to us as cardiologists is the 22q11 deletion. There's deletion on the region 11 on the long arm of chromosome 22. 1 out of 4,000 patients will have this deletion. This has been reported in various syndromes, including the well-known DiGeorge syndrome, the velocardiofacial syndromes, the conotruncal anomalies, and others. And please note that patients who have this particular deletion will have congenital heart disease in about 85% of the cases. Patients who have, for example, tetralogy of Fallot that's associated with no particular chromosomal abnormalities but associated with some syndromic features will have this deletion in about 15%. Also, some cases of conotruncal abnormalities with no apparent dysmorphic features are reported to have this particular deletion. This deletion involves about 30 genes or more, and the base pairs may be about 3 million base pairs or more, and it is associated with one of the transcription factors, TPX1, which we will discuss in more details in the upcoming slides. Please note here that the deletion is present in this area where we do not have a fluorescence while this particular allele is a normal allele where we have the probe attached to the gene and appearing in this particular color here. Another syndromic features are associated with the Williams syndrome. These patients have typical dysmorphic features, have supravalvular aortic stenosis, branch pulmonary artery stenosis, and have deletions in the on chromosome 7. Again, the elastin gene in these patients is not present. This is a patient with normal chromosome 7, while if we look up here, this is the elastin gene present in one of the alleles. While using the FISH technique, one will not be able to detect the fluorescent, and thus this patient has deletion on chromosome 7, which is associated with Williams syndrome. Again, another disease where we have the Creux-de-Chat syndrome. These patients have typical features abnormality. They have typical cry, described to be as a cry of a cat, and they have deletions in chromosome 5. Again, we have the chromosomal abnormalities which can be detected by karyotype. We have the gene deletions which can be detected by the FISH technique for particular syndromes. And last but not least, we have certain abnormalities that involve certain syndromes where the problem involves mutations of certain nucleotides or acid pairs. These cannot be detected by the regular karyotype or by the FISH technique. As we said, FISH technique may involve large number of genes that may include a 1 million base pair. But if you have deletion in certain nucleotides, you have to use the PCR and the sequencing technique. And one of the known syndrome that's associated with mutations in particular nucleotides is the Marfan syndrome. These patients have certain mutations in certain amino acids, and these can be ordered from special labs that are present in few areas in the world. Also, the well-known Holt-Oram syndrome. These patients have, as you well know, VSD or ASD in addition to skeletal abnormalities. It's an autosomal dominant disease, and the deletion has been described lately to be, the mutation has been described lately to be on chromosome 12. It is triggered by a transcription factor called TPX5, and this transcription factor, which is a special protein, will encode the TPX gene which is present on chromosome 12. Again, this can be detected by doing a regular PCR and DNA sequencing for the involved axons of the genes, and they are available in very few labs in certain centers. The Chart syndrome is also have been reported to involve mutation in the TFAP2B. This is a gene present on chromosome 6. These patients have typical syndromic features in association with a patent ductus arteriosus. The well-known Alagille syndrome is also another example where mutations in certain nucleotides is reported to affect the JAK gene. This is present on chromosome 20, and again, this particular mutations can be done to confirm the diagnosis in special labs that are present and do it commercially. Also, mutations have been reported in 1 or 2 familial cases of the heterotaxia syndrome. And of course, the well-known association of mutations in certain genes with the entity of cardiomyopathy, in particular the hypertrophic cardiomyopathy, which has an important role in the prognostic and diagnostic factors of this anomaly is